While we introduce artificial regulation of enzyme activity with inhibitors, our cells need to be able to do this naturally, because sometimes there's not enough substrate for our cells to work on, or for specific enzymes to work on. Our cells need to be able to turn off that enzyme when there's not enough substrate, and turn it back on when the substrate comes back. Or if there's too much product, for example, if we have too much ATP, our cell needs to be able to tell its enzymes to slow down and use whatever we have to make something else instead of ATP. We can do this within our cells in 3 different ways. The first is allosteric enzymes that are under feedback control. In a very broad sense, this is when we have a molecule that either turns on or off enzymes. We'll talk more about this in the next slide. Enzymes also can be activated by other enzymes. This is really helpful when our enzymes do something that we don't want them to do. We need to turn them off initially and turn them on later when they're in the right environment. Finally, we modify our enzymes covalently. We add another group on, a prosthetic group, oftentimes a phosphate that's added using ATP or an oligosaccharide. In each of those cases, the phosphate or oligosaccharide are really polar and can change the overall structure of the enzyme, either turning it on or turning it off. Allosteric enzymes, again, enzymes that are able to be regulated by other molecules, have a couple of interesting characteristics. They almost always have quaternary structure. They have separate binding sites for substrate and regulator. So notice there's a binding site and active site, and there's also a site where the regulator can bind on. Notice the regulator might be negative or positive. So when a regulator binds on, it will change the protein structure, and it might make it so the active site is no longer there, make it slower, or it might make it so the active site is actually better. Both can be true with allosteric enzymes, which is different than inhibitors. Inhibitors will only slow down the rate of an enzyme. Allosteric regulators can either be negative and slow down the rate of an enzyme, or they can be positive and speed up the rate of an enzyme. Feedback control is a specific mechanism where we use a product of a reaction to slow down an earlier enzyme. This can be really helpful in a metabolic sequence. So for example, if we're taking glucose and eventually making ATP, we might not want to use up all our glucose if we have a lot of ATP. So think of this product D like ATP. If we have lots of ATP, that ATP can come back and bind on to enzyme 1, telling enzyme 1 that it needs to slow down. The enzyme 1 then will be able to slow down as it waits for the ATP to be used up for other reactions, keeping the glucose for later. Our second example of ways in which our cells regulate protein activity or enzyme activity is by using proteolytic enzymes. Proteolytic enzymes are enzymes that break peptide bonds. Usually those are really dangerous to have produced in our bodies in an active form, because the same place that those proteins are produced are where all the other proteins are produced. And if they break peptide bonds, break proteins apart, we would produce all sorts of proteins and break them apart all at once. Proteolytic enzymes then are produced inactively. They're moved into the lysozyme where all proteins are broken down, and once they're in the lysozyme, they'll be converted to an active form by breaking out a protein fragment. So this zymogen, or proenzyme, the inactive form of the proteolytic enzyme, has this peptide fragment that keeps it from actually functioning. As soon as it enters the lysozyme, that piece can be cut out, and now the protein is going to fold the right way to start cleaving other proteins. Covalent modification is the last way where our bodies and cells are able to regulate enzyme activity. This is accomplished by attaching some sort of prosthetic group onto the enzyme. Usually that attachment is reversible, although it's covalent, so it's relatively permanent. The most common form of covalent modification is phosphorylation. Phosphorylation will take phosphate from an ATP and move it onto the enzyme. Another enzyme will often catalyze this. Those enzymes are called kinases because kinases are the transferases that move phosphate from one molecule to another, in this case from ATP onto an enzyme. Again, Covalent modification can change the rate of an enzyme either up or down. It depends on the enzyme.